It gives me a great deal of pleasure to be here today. I know that the common thread amongst all of us is the steadfast belief that genomics will drive improvement to the overall patient outcome experience and to cancer patients. It is that mission that drives us forward in transforming Illumina from traditionally a life science company to a healthcare company. That is a painful and costly experience, but it's one that we take on with a full degree of vigor. It requires the work and the effort and the commitment of some extraordinary individuals that I have the pleasure of calling my teammates. As part of that, our plan is to bring to the market in vitro diagnostics to standardize genomic testing across the industry. It is then our plan to open those platforms to ensure that they deliver all the solutions that you want and that you can customize those solutions to your care. And then we want to repeat that and tackle as many high-value applications as we possibly can to keep driving the science forward. And that's exactly why we have partners like you here, so that you can teach us and tell us what we need to do differently, what else we need to do to keep driving this mission forward. So I'll stop right there, and I'll introduce our speaker. Eliezer Van Allen comes to us from Harvard. Let me bring up his bio here real quick. So he's an assistant professor of medicine at the Harvard Medical School. He's a clinician at Dana Farber, and he's an associate member at the Broad Institute of MIT, so complete underachiever. His research focuses on computational cancer genomics, as well as personalized medicine, and specifically around mechanisms of therapy resistance. He's originally from Los Angeles. He studied symbolic systems at Stanford University. And I had to look that up because I actually thought that was some sort of like a Swedish film kind of program. And he got his MD from UCLA and completed his residency in internal medicine at UCSF. So with that, I'll turn it over to Dr. Eliezer Van Allen. Thank you so much.
Great. So thank you for the introduction and for giving me this chance to present. It's a privilege to be here at this function, in part because I'm sort of a downstream consumer of so much of what you guys do here, and it's been sort of instrumental for my academic career. So as was described, so I'll give a sort of try to give you a global overview in terms of what we're doing in the space of genomics and immunotherapy, given sort of the sort of transcendent properties of each of these as they're applied to clinical oncology. So in terms of my outline, I'll go into computational oncology and precision cancer medicine in general, and then describe really how that's actually sort of converge with what's happening in the world of immunotherapy and immune checkpoint blockade specifically for patients with advanced disease, and then look into how we're not only looking at using this really for understanding why certain patients respond to these agents, but also really to understand sort of how these patients become resistant. And I think that's an emerging story that actually hasn't sort of gotten as much attention as it probably will over the next 1 to 2 years. But even for the folks that do benefit from these really transformative drugs, more often than not they will become resistant. And we need new techniques and tools to understand why, so we can come up with the right combinations moving forward. But of course, this is always the case. And for this event and for every event, I'm obligated to report my personal disclosures, which are here. I got to add the Illumina speaking one for this event. But I should also say I'm a computational biologist, so naturally I should mention that I am an equity holder in Microsoft. I'd like to thank the Gross family for giving me what turned out to be easily the best gift I got for my bar mitzvah. But I'd also use this as a pivot point to actually sort of flesh out a little bit more as to sort of how I ended up getting into this space. And I think this is a little bit different than sort of the average academic oncologist, but may sort of hopefully give you a window into sort of how I ended up doing this and perhaps how we can all work together to sort of solve these bigger problems. So there's me with a lot more hair and a lot darker hair. So as was mentioned, I went to Stanford and studied something called symbolic systems. I graduated in 2003. So this degree program is actually a mix of computer science with philosophy, psychology, and linguistics. All of my friends are multimillionaires because they all went off from these degrees to work at the time, startup companies, Google, Facebook, and so on. And actually, the reason I ended up going into medicine was almost by accident. So I got involved to help create this thing called Camp Kesem. It's a camp for kids who have or had a parent with cancer. We created the first camp in 2001 out at Stanford. And I will just say, in all honesty, it was a transformational experience for me. I'd never actually done anything quite like this. I'd never really experienced sort of interacting with like kids who were dealing with cancer, their parents who were dying of this disease, and it sort of completely changed my life. And that's actually why I went into medicine, and specifically into cancer medicine. But if you fast forward from that sort of really sort of inspirational experience and sort of what's happened since then, and I'll just say sort of tangentially, we started with one camp in 2001. There's now about 100 of them around the country, and I'm on the advisory board for the one based out of MIT, which is pretty cool to be a part of. So that experience notwithstanding, I sort of ended up sort of going into medicine and having a little bit of trouble figuring out where I belong in this space. And it's really only sort of recently that it's emerged to me, it's become real to me that there's this huge opportunity to blend the technology and sort of the work that's happening with Illumina and really sort of frankly, everyone in this room with everything else that's happening in clinical oncology and sort of towards a new field almost. So the field that I've sort of created for myself is sort of as follows. So I'm a clinician. I see patients every Monday morning except for today because I'm here over at Dana Farber in the GU Oncology group. I'm a computational biologist by sort of fundamental training through both my undergraduate experiences and my postdoctoral fellowship with Levi Garraway and Gady Getz at the Broad Institute. And with those experiences comes sort of a wealth of cancer biology that I've got exposed to. And as a result of that, I sort of ended up creating a field for myself, and this is what I call what I do because I really don't know how else to describe it, a clinical computational oncology. So the patients are my model system. I tried my best to sort of be interested in cell lines and mice, and I just couldn't do it. But patients are fundamentally the folks we're trying to learn from, and that I had an opportunity to do that because I see them. I'm a clinical oncologist. I'm a computer nerd, and I can sort of, as a result of the technologies from Illumina and elsewhere, I can actually generate increasingly vast amounts of data from these patients and develop algorithms to make sense of it all. And I can put it into context. What is happening in the world of cancer biology? How can I actually use that to make sense of this information? And that sweet spot, sort of right in the middle of these intersecting circles, is really what we do. And that's what my lab does. And that's really sort of how that guides everything that we think about moving forward. And it was sort of convenient that this was happening for me right around when this was happening. So precision medicine, and for the purpose of this talk, for me, precision medicine is the use of multi-omic technology and large-scale data to guide individualized patient care and make new discoveries that can actually inform new targets and new processes that we can actually use to help patients with cancer. And of course, and it goes without saying for this group, I would imagine, but it's really worth repeating that this idea of precision cancer medicine truly is a potential paradigm shift. Potential being because, we all know we haven't really sort of gotten all the way to the finish line yet by any means, but the idea is a pretty simple one. If you have advanced cancer, we can test you for certain kinds of genetic targets based off of what your tumor looks like. There's an increasing number of those from sort of the more canonical targeted therapy perspective, and match you based off of your genetics to the right drug. And this has sort of been a principle that was pushed forward really when I was starting my fellowship, 2010, 2011. But to be perfectly honest, and sort of to stay true to the title of this talk, that paradigm feels a little dated. The world of sort of like single genes, single kinase, sort of we're going to solve all cancer, we thought might be the case in 2010, by 2011 or so. But we all know now that it's sort of course, the story is more complicated. But what was happening while we were developing these technologies and these approaches is this revolution in immuno-oncology. And to illustrate that, I actually sort of have this nice table, and this is a summary table from Kathleen Mahoney, who published a review on this a year ago. She's a colleague and friend at Beth Israel. This was sort of a table trying to summarize really what some of the immune checkpoints were. And I illustrate this for a few reasons. So one, you can see sort of where CTLA-4 and PD-1, PD-L1 are, but more important than that is actually, this table goes on basically all the way to the floor. I mean, there's so many immune checkpoints in development in various ways, shapes, and forms that you could not possibly squeeze it all into one slide. And frankly, as I'm sure you guys have now seen as a result from this meeting alone, you probably you can spend a whole talk just reviewing all the different immune checkpoints that are in some sort of clinical development. And when we were looking at this, we said, gosh, I wonder whether there's a way to actually make a convergence of what we're doing in the genomic space with what's happening in the clinical oncology space, and apply these principles of precision cancer medicine, whereby we encounter patients, we do omic profiling, we try to match patients to drugs, we follow these patients over time, we study these patients as they become resistant to these drugs, could actually be applicable to immuno-oncology in the way it's been applicable to targeted therapies and chemotherapies. And with that as a backdrop, what I want to do is actually give you a couple of vignettes into sort of how we're actually thinking about this now. So one example of this is sort of how we use genomics in the space of immuno-oncology is really understanding sort of what are potential biomarkers of response, a selective response to immune checkpoint blockade. Who's going to respond and who's going to not respond, given a population of tens, hundreds, thousands of patients. And that's really sort of the fundamental question we've been trying to ask for the most part in this space of clinical computational oncology as it's converged into the world of immuno-oncology. To do that, actually, I want to walk you through this study and sort of illustrate why we're at where we're at and where we have to go. I'll sort of first actually just describe sort of the study schematic. So this was actually work that we did in collaboration with Levi Garraway, Dirk Schadendorf, and his team out in Germany, the DCOG, the German cooperative group, whereby we actually were able to consolidate and collect a cohort of patients with metastatic melanoma, all of whom had fresh biopsies and then subsequently went on to CTLA-4 monotherapy within 3 months of the biopsy. And I think before I even sort of say anything further, it really is to the profound credit of our colleagues in Germany that they were able to abstract the clinical data for these patients and tell us who were the responders, who were the non-responders. Because as those of us who are practicing physicians in the United States know, it's actually frankly trivial to sequence a genome at this point, courtesy of all of your guys' hard work. But figuring out who responded and what drug they got is a profoundly epic problem. Sorry, I'm really sorry. That was a terrible joke. But for those of you who are in the know. But needless to say, that's actually the rate-limiting step now for discovery. It's not the genotype, it's the phenotype. But in this case, we were actually able to get that information. And we were able to not only generate whole exome sequencing data of the tumor and germline from these biopsy samples, in a subset of patients we were able to apply capture-based transcriptome profiling from formalin-fixed paraffin-embedded tumor blocks using this technology to really try to get a more holistic picture of all of the different things that can contribute to selective response. And I'd say this is, thankfully for me, this is work largely done by Diana, a medical student in my lab who, unfortunately for me, is now actually going back to medical school to finish off her training. But needless to say, she was able to apply sort of the latest and greatest algorithms to these large-scale data to try to make sense of it all. And I say that because, there's a lot of cool stuff we can do with this data in this space of clinical computational oncology. When we have exomes and transcriptomes from enough patients, in this case over 100, we can ask questions about mutational load, neoantigen load, expression of certain neoantigens that may be relevant and provoke an immune response. We can start to explore sort of really more profound case-controlled genomics and actually try to look for single gene biomarkers of response. With the transcriptome data, we can actually look at pathway activation. We can start to look at immune infiltrate in the microenvironment from these bulk tumor samples, and not from very selected frozen tissue or fresh biopsies, but again, formalin-fixed paraffin-embedded samples, like the stuff that really constitutes 95% of sort of at least the United States' pathology samples. We can actually do this on. And just to sort of pause for a second so you know what I'm talking about with respect to neoantigens, I'll try my best to summarize all of immunology in 30 seconds or less. So the simple dogma is if you have a patient who has a gene, DNA, RNA, protein, gene goes to a protein, and what happens in addition to whatever that protein is supposed to do is that the protein will get chopped up into 9 or 10 amino acid length strings. And those strings will get presented on the cells of every cell in the patient's body, and the immune system will surveil that and sort of pluck out things that might be sort of damaging foreign bacteria, those kinds of things. In the normal setting, everything is sort of sensed as self, and those are antigens, and everything works fine. But of course, in cancer, genes get mutated; those turn into mutated proteins, and those proteins can accidentally change the sequence of these peptides, these 9 and 10 amino acid length strings. and they can create things that are so-called neoantigens. And it is conceivable that these neoantigens may appear foreign to immune cells in the right environment, and perhaps may actually provoke an immune response if the brakes are released as a result of these immune checkpoint blockades. And that's sort of the principle behind a lot of why these drugs are in the development. For those of us who are computer nerds, who don't even want to worry about biology, you can start to just imagine looking at these strings of letters and start to say, gosh, I wonder if there are patterns in here. If only I had enough data to actually make sense of these patterns, I could actually train a computer to figure it out for me. And even that is actually starting to really push us to think about what does it actually mean to be a cancer driver. We've spent the last 10 years through TCGA and all sorts of other efforts really thinking about the drivers of oncogenesis, KRAS, BRAF, EGFR, and so on and so forth. It could be any random passenger mutation that just so happens to make one of these funky neoantigens that can provoke an immune response, and that could become a sort of quote-unquote driver in the setting of the right drug. So we built algorithms to look for these things and sort of summarize sort of the big picture of what we saw. So when we broke patients up, again, using the sort of the very helpful and rich clinical annotations from our friends in Germany, by no clinical benefit, and this subset of patients who had long-term survival with sort of quote-unquote no clinical benefit because they were on drug for a short period of time, we could see that mutational load, sort of just simply counting the overall number of mutations, was correlated with response. Similarly, deriving in silico putative neoantigens from those mutations, from those somatic events, no matter where they came from, and matching those to the patient's HLA, counting those also, you could yield a p-value that's significant and correlated with response. And we saw this in our study with metastatic melanoma and CTLA-4, but we know we're not the only ones that saw this. We saw this in melanoma. There was a group at Memorial Sloan Kettering that saw the same pattern. That same group saw the same observation with respect to PD-1 blockade in non-small cell lung cancer. A different group at Hopkins found the same observation with PD-1 blockade and microsatellite unstable tumors that are often hypermutated in colorectal cancer and elsewhere. And the constant theme here was mutational load. Simply the more mutations you have, and thereby we think the more neoantigens you create, the more likely you are to, by accident or otherwise, have a peptide that might, in the right setting, provoke an immune response. And all of that data is actually derived from the genomics we were getting that we would have never necessarily imagined using for this purpose 3, 4, 5 years ago, but are now doing on a regular basis. So much so that there's a lot of interest in actually taking this observation and plopping it right into the clinic. And we know that it's more often the case that clinics don't typically do clinical exomes, they'll do panel testing and whatnot. So we asked a simple question in a follow-up study, how small of a panel can you get where you can still actually infer the global mutational load? Here's an example of taking 300 genes. This is a 300 gene panel from Dana Farber's profile project, where you could actually pretty reliably predict the global mutational load from a small panel set. Not surprisingly, the smaller the gene, actually before I get to that, it actually was pretty fun. You make a simple equation to actually do this. And I presented this at ASCO last year, and to the clinicians in the room, in the audience, they just started, you can see the beads of sweat sort of roll down their forehead. It's like, oh God, like he's doing math. It was interesting, the panels overestimated the loads by a constant variable. It's not surprising. We're enriching these panels for genes that are mutated in cancer, so of course we're going to overestimate it. It's also not surprising that the smaller the panel, the less likely you are to actually predict this mutation load. So this is relevant because we actually pulled a couple of commercially available panels, including some from Illumina, where it's the very small gene panels cannot predict mutation load. We kind of figured that was the case, but again, this has relevance if we're going to start to use this at the point of care. But that's actually the key point, and I was just chatting with a colleague and a sort of a researcher in this space, Jesse Zaretsky, about this just now a few minutes ago, whereby you could look at this same figure and very appropriately say to me, like the thing that I said to myself for the last 2 years as I was staring at these data, and say, okay, mutational load and neoantigen load correlate with clinical benefit. That's great. But I mean, come on. Look at all those high mutational load non-responders, and look at all those low mutational load responders. How good of a clinical biomarker is this, if at all? Why is that the case? What about class 2 neoantigens that we're not even considering? shouldn't we be getting larger clinically annotated cohorts to actually drill into this more and come up with really sort of better, more clinically deployable solutions and or sort of actual targets that we can go after and not sort of vague instances of mutational load? One way we actually can try to infer, sort of make a better model to help us predict sort of who's going to respond and who's not going to respond is consider not just the mutations in the tumor themselves, but also the immune system and the microenvironment. So I say that, and when I say something like that, the immunologists in the room sort of like look at me with their eyes rolling in the back of their heads because they said, of course, that's the case. But I came at this from a very tumor-centric genomics point of view. But thankfully, we can actually start to measure that using these same technologies. So, for example, there was this beautiful 2-gene signature of cytolytic T-cell activity derived from Neera Cohen's lab over at MGH that was used across the Cancer Genome Atlas to really infer sort of immune infiltrate and bulk RNA sequencing data. And we took that very simple 2-gene signature, had generated transcriptome data from our patients in this study that I described, and could also show that this cytolytic signature correlated with a clinical benefit to immune checkpoint blockade in this cohort as well. Of course, the same caveat that I mentioned to you earlier about mutational load applies here. This was not profoundly significant and on its own was probably insufficient to explain response, but you can start to get a sense of saying, maybe there's actually a way towards a predictive model I can build with whole exome sequencing and whole transcriptome sequencing that can help me segregate which patients are actually going to benefit from these drugs and which are not. I will say, so right now, these are still, I would say, in the early days of trying to infer this. There's a lot of technical challenges and other kinds of considerations. But needless to say, we think this is a promising path forward. In addition to sort of doing these things at a larger scale, we also want to think about what we can learn from these outlier cases that really may help us sort of get a sense into what's going on, not so much from a global mutational load perspective or sort of in a very vague sense what's happening in the immune microenvironment, but are there specific genes that matter? And so here's an example of one patient. This patient had non-small cell lung cancer, was about to enroll in hospice after becoming resistant to standard of care chemotherapy. There's a sort of a very non-subtle large mass starting to obstruct the trachea with a big red arrow. The patient happened to be very weakly PD-L1 positive, was enrolled on a PD-L1 inhibitor phase 1 trial, and very quickly had a response that was profound. The tumor disappeared. It was off. It basically was on the trial for a year. The study ended. Was off the drug for 18 months. Had a recurrence. Was rechallenged with the drug and is still alive and well now. So now I think we're 3-4 years out. This is metastatic lung cancer. That's not normal for those of you who are not clinical oncologists. And a long story short, what was kind of fascinating about this was there was actually not only a somatic point mutation in JAK3 in the tumor. There was a mutation of V722I. There was actually a germline polymorphism that was present in the tumor. It was present in all the cells of the patient because, again, it was germline. And that actually is a known activating polymorphism of this gene present in less than 1% of the population. And then all of a sudden you're seeing, oh gosh, activation of JAK-STAT signaling in this patient who has sort of had a supercharged JAK-STAT signaling not only in the tumor but also in the immune cells and everywhere else, was associated with this exceptional response. And I think that's especially exciting given that we're now seeing evidence from Tony Ribas's group and elsewhere that loss of function of these same genes associates with resistance. And so we're starting to put this puzzle together with more and more clinically annotated molecular data from patients getting these drugs. In terms of other specific genes and response, this is just a study that we sort of put in our supplement of our paper because we basically didn't find anything. That's in large part because we were underpowered to answer the question. We think we're getting closer now that we're getting sort of an order of magnitude more samples with clinical annotation to actually figure out which genes, which signaling pathways matter, because that's relevant for drug development and whatnot. But needless to say, we're working on this and hoping to work with sort of everyone in the community to answer that question. But again, I sort of painted a picture about how we're using these data sets at one time point to try to come up with these models, recognizing that now we're in the world where a lot of these patients have gotten these drugs and are starting to become resistant. So just in the way we applied the sort of serial biopsy paradigm to the targeted therapy world to understand what the genetics are of patients who become resistant to targeted therapies, it was really transformed by the genetics that we were able to obtain from these biopsies. Could we use the same thing for patients getting immune checkpoint blockade. In essence, if we follow these patients over time, we know that there's going to be variable responses, but, and some kinds of exceptional responses, but can genomics explain clinical resistance to immune checkpoint blockade? So here's an example of a study that came out of our lab. This is a pretty exciting case. So this is a patient with a metastatic uterine leiomyosarcoma. This is a mesenchymal tumor. It's a rather rare disease. This patient, to our understanding, we think may be the only patient with a leiomyosarcoma who's ever responded to immune checkpoint blockade. And sort of for non-subtle sake, the large lesion in the liver was sort of one of the primary metastatic deposits after the patient's uterus was taken out. And the patient was enrolled prospectively on a phase 1 pembrolizumab trial, had a profound response to that lesion and to every other metastatic lesion in the patient's body except for this one lesion in the pelvis that kept growing, the C to F panel in this figure. And after about a year on drug, after every other lesion had basically shrunk to nothing, the clinician said, maybe we should just cut this one lesion out and cure the patient. So they took the patient to the OR, cut that one growing lesion out, and all of a sudden we were armed with a pre-treatment biopsy in a patient who had a profound response with the exception of one small focus that kept growing and could apply this kind of paradigm to this patient's data in a rare cancer that, for the time being has no clinical development of these drugs, but maybe should. And we can, in this case, not only do longitudinal genomics, but integrate this with immune profiling, immunohistochemistry, and other things that we think are relevant for immunotherapy that may perhaps have not been as relevant for targeted therapies or chemotherapies. And so I'll just show you the results, and this is something I just published less than a month or so ago. So this is how you interpret these plots. So on the x-axis is what's called the pretreatment samples cancer cell fraction, or CCF. And this is a purity and ploidy corrected allelic abundance of every variant. So to deal with the confounders where sometimes one biopsy is more pure than another, and when you're trying to understand whether there's changes in an allelic fraction of a mutation, you can apply computational techniques to actually make sense of it all. So on the x-axis is the allelic abundance, or cancer cell fraction, for the pretreatment tumor. On the y-axis is the same thing for the resistant tumor. And so basically on the y equals x line are things all the way on the top right-hand corner of this plot are mutations that are clonal in the pretreatment tumor and clonal in the resistant tumor. Things on the top left of the plot, so top right of the plot was clonal in both. Top left is clonal in the resistant tumor and not detected in the pretreatment tumor. And bottom right is clonal in the pretreatment tumor and not detected in the resistant tumor. And you can use this information to not just make these fancy plots, but also actually reconstruct a phylogenetic tree of the evolution of this tumor over time in the context of therapy. And that's what this figure on the right denotes. Historically, what we've done for all of our targeted therapy resistance studies is our eyes go straight to the top left of this plot. We say, what happened? What changed? For instance, with BRAF mutant melanoma, is there an NRAS mutation? Is there a MEK1 or a MEK2 mutation? And is that why the patient became resistant? And that rule applies here. But what's equally relevant here, again, going back to this paradigm of neoantigens, is asking the question of, well, were there mutations in the bottom right of this plot in the pretreatment responding tumor that I don't detect in the resistant tumor that may have accidentally, by some random chance, created an immunogenic peptide that could provoke an immune response that disappeared, or that's just for whatever reason, and I don't want to go too far and speculate as to why, or not detected in the resistant tumor, and could that be contributing to response as well? And that's what we did. So in this tumor, the first sort of notable result was there was clonal p53 mutations in both the pretreatment and resistant tumors. That's not helpful for us. But what was really striking was that in the top left of this plot, there was literally only one non-synonymous mutation in the tumor that was unique to the resistant tumor, which was the second hit in PTEN. The reason why we thought that was interesting was that literally, as we were sort of deriving that data, there was a report from MD Anderson showing biallelic loss of PTEN contributing to acquired resistance to immune checkpoint blockade in melanoma. And now we're saying, maybe this is actually relevant to this one patient, this outlier responder who otherwise was having a profound response in leiomyosarcoma as well. And we think that's actually contributing in part to this story, but again, We also want to think about what's happening in the bottom right-hand corner of this plot. What are the neoantigens that may have come and gone? And in fact, because we had both whole exome sequencing and whole transcriptome sequencing from these paraffin blocks, we could actually measure, derive not just the neoantigens we saw, but also the relative change in expression of these neoantigens. Were there some that were, we think, quote unquote, downregulated? We don't want to over-speculate on sort of given the technical issues with these data, but needless to say, there were a few neoantigens derived from point mutations in genes that have otherwise no known function in cancer that seem to be downregulated selectively in the resistant tumor compared to the pretreatment tumor. And what we did next was we said, that's pretty cool. We can actually synthesize these peptides, or get them from any company, and this patient is alive and well and just happy to be here, or not here literally, but you know what I mean. She just came by, gave us some more blood samples, and we can just take her T cells and say, let's just test these peptides against her T cells, figure out which ones provoke an immune response. And that's what we did. And we found that actually the two with the steepest drops in expression, the mutated genes that created neoantigens that had the most downregulation of expression, it's a bit of a mouthful, hope I'm explaining that correctly, were also the ones that provoked the highest immune response to the patient's own T cells. And all of a sudden, we can go from where there's just way too many of these putative neoantigens to ever make sense of this to, in at least one patient, drill down to what we think are the only two that really mattered, the two that disappeared from the resistant tumor. So that's in essence the sort of broader framework of how we're trying to apply this, whereby we identify patients going onto these drugs, we try to match them to the right immune checkpoint blockade, or to any immune checkpoint blockade at this point. We follow these patients over time. We build increasingly large cohorts of responders and non-responders, where we can generate these larger sets of clinically annotated molecular data so that we can actually feed this information back in towards building a predictive model of response that can actually help guide us clinically, figure out who should actually try these drugs and who shouldn't. We know these drugs are toxic. They have some very severe toxicities, and maybe not everyone should benefit, or should get it rather. And likewise, this will hopefully help inform for us what are the right combinations, which other signaling pathways are invoked as a result of these therapies, and how can we use this for drug development, target discovery, and clinical trial design. So thank you again for your attention. Again, I hope this was sort of informative for this group. Obviously, a lot of this work is really to the credit of a huge team effort across my lab, the Broad Institute, Dana Farber with Levi, who's no longer here, a Stand Up to Cancer team, the country of Germany, and I'll just say mostly thanks to my wife, who's dealing with those two monsters in the top left corner of this plot, all by herself. If anyone knows any nannies who are looking for work in the Brookline area, please let me know. We are suddenly out one, and thank you very much for your attention.